my question is about the limitation of galaxy.
Okay.
Yeah.
Okay, so thanks very much once again, James. Just to let you know, I hope you don't feel that I'm hurrying the speakers, but we only have 20 minutes for each section, so I'm trying to sort of make sure we keep our time. And that brings me to our second speaker for today, who's Mike Sternberg from Imperial College in London. Mike's going to be talking about the Phyre2 protein modelling analysis made easy. So, Mike.
Thank you very much. So it's a talk mainly about Phyre2, which is a protein structure prediction server, but we've also got something to present about an easy tool to molecular display, and if we have time, some fun games on protein modelling. So Phyre2. So the idea is protein structure prediction. From sequence, you've got a sequence, you want to know the 3D structure. It's based on the method of template-based prediction, so you have to recognize an homologous protein. And the latest publication is in Nature Protocols 2 years ago, and the person really who's been the brains behind this is Lawrence Kelly. So here we see two homologous proteins, 25% identity, very similar 3D structures. So in a sort of simplified version, how does template-based modeling work? You recognize that the two proteins are similar. You adjust the main chain to represent the new sequence. You stick on the side chains, and hey presto, you have your predicted 3D structure. So that's a sort of simplified view of the strategy. So now, how does Phyre work? Well, the first thing is we want to have a very clean interface, so you simply have to paste in your sequence, give an email address, and then you can hit run. So there's no complicated options in the beginning that terrify people before they even start the analysis. It's proved highly popular. It's 4,000 or 5,000 citations to the papers, and we've had over 1.5 million submissions. The main reason is that Lawrence misspent most of his youth doing computer games and tended to make the interface attractive for users. And now I hope to show you why people like it. The results, typically you get them back in 2 hours. And there are other facilities that you can do batch mode. You can put loads and loads of sequences. So the driver has been to make it useful for the biologists. So the first thing is to recognize that your query protein is similar to a protein in the database. And here we use now the most sensitive approach, which is the statistical hidden Markov model. And so you have on the left a hidden Markov model of your query, and on the right a member of the database with its hidden Markov model. And in addition, you have the predicted secondary structure and the observed secondary structure. So you're matching sequence and some information about secondary structure, and hence you can call this fold recognition. So technically, you would put in your query, we run Serding's H8 splits, gives us a multiple alignment. We then run David Jones' Cypred secondary structure prediction. Combining them, that gives us our query. Previously, we have set up an HMM database of the known structures. You match one against the other, and you get an alignment. So you've recognized the relationship, you've generated an alignment. And then the next step is to start from the alignment, get a crude backbone, add in the regions that are variable, typically the loops, add the side chains, and then you get the final model. So the loop modeling is the traditional spare part approach, that if you need 5 residues to fit, you search the protein data bank for fragments that match the takeoff positions and have the right separation, and then you carry that forward into there. It's a sort of Frankenstein approach. We're building bits together to make the entire protein. And then you use a library that indicates the likely positions of the side chains and finds the optimal packing. And we're now going to implement Dundrecht's squirrel approach. And so that's the strategy. So you've put your query in, and then you get a series of results. On the front page, you get the top model. And here you'd have an image, and you get the confidence that you found the correct template, how much of your template is covered. Down here is something called fire alarm. I'll describe that shortly. And you can click on the information and find your template. So the top page gives you your best solution. Then you can drill down and you can get a prediction of secondary structure and potential disordered regions. Then you can drill down and, in fact, not the entire sequence might match, but you might get different coverages for different parts. So over here, here's your best match, but locally there can be some fragments of other domains that can match. And if you weren't to have this top hit, to get a 3D structure, you would have to meld these three components together. And that melding is done when you put in the intensive mode. So there's the normal mode, which uses a single template and is more accurate, but when you need to combine domains, that's the intensive mode. And then you have the detailed template information. So you have a range of templates. You can click on this and download the coordinates. So it's not just an image, it's a model. And you have the confidence in your model. You can check the alignment in detail. And over here you see there is something called Phyre Investigator. And the alignment view enables you in detail to match your query with the template, see which residues align, and do you align, for example, functional residues, which might help you to choose the correct template. This is the intensive mode. So here you would have found models for different parts of the protein, here, and you're linking in a fragment-based approach the connection, and we use a simulation called Poing, and you get a final model. That gives you some indication, but you really can't rely on the relative positions of the two domains, except if the connection is short. So how accurate will your modeling be? It depends on the sequence identity. If it's very close, you typically will be about 1 angstrom. As the sequence identity decreases, you have longer loops to model, typically give you 5 angstroms. Below 20, the alignment can be poor, and then you can start getting large errors. Additional features, there's Phyre Investigator. This enables you to drill down and see which parts of the model are reliable, and also we've incorporated a variant prediction which we suspect from our group, and that will tell you as you go along the sequence which residues are likely to be, if their amino acid is changed, have a major effect on the phenotype. So this is the Phyre Investigator. You can drag along the sequence. You see where the region is. This gives you an idea of the confidence. People just don't want to predict the structure. They won't want to look at a binding site. And you can say, yes, this is the region I'm interested in. That's well predicted. And then on the right, we have the suspect score. So you can see down here that if that's the substitution, that's likely to be deleterious. Suspect, there are many variant servers. As more and more sequencing, they're sort of coming out rapidly. Sequence conservation is standard. That's the sort of SIFT approach. Structural features typically incorporated in polyphen. What we've added in is protein-protein interactions. The proteins that tend to have many interactors tend to be more susceptible to disease, and that's an added feature in suspect, and that in our benchmarking led to added accuracy. Now, the protein data bank is increasing every week. So we don't envisage our users come in on Monday morning and say, I ran it last week, and I wonder, before I have my cup of coffee, I'll rerun Phyre because maybe there's a new protein data bank. So we solve that for you. You just hit fire alarm, and every week we will find if there's a new match in your structure, and if it is, and it's important, it will be returned to you. So we will do the work for you. So this is the sort of sequence, and every week we will try again, and you will get the result back if there's a better model. Because as structures increase, you can suddenly find 6 weeks down the line, you've really got a good model. 3D ligand site from MarkWass. This incorporates the idea is where a ligand is likely to bind on your predicted structure, so you can go from Phyre straight into this prediction, and that is based on using homologous proteins and finding clusters of ligands. So in the same way that Phyre uses homologous structures to build the model, you can also use homologous structures to identify where the ligand binding site is. I hate computers, and I'm also getting old. And so whenever I run Phyre and I want to look at a structure, I download it, I look at it, and then I say, I want to color residue 46 because that's where the variant is. And you pick up Pymol, which is superb, and then I say, I don't know how the hell to write the command, and I run to my computer manager, help me. So I thought that's not really a good scenario. So over the last couple of years, we've developed something called Easymode. And this is a bit like EasyJet, nothing flash, but does what you want. So the idea is designed for the occasional biological users and professors. It works over the web, so there's no download. Sometimes people can't get access to their computer. The computer manager stopped you doing anything. Very limited set of options. It's wizard-driven tabs. Few mouse clicks, you get to what you want. No command lines. You don't have to do select 15 semicolon asterisk blue. No need to read a help file, hopefully. You can produce images for publication, and you can save your work. So there's WYSIWYG. What you see is what you get. This is what you see is what you need. So I've tried to take in what the sorts of representations I feel people might want, and then developed a limited set. If you want to do brilliant molecular graphics, use Chimera and Pymol. We're not in that league. But if you're like me and can't remember a command line from one week to the other, try this. So this is a movie, hopefully. So you have to type in the PDB code, either from a file you've got or from the data bank. It comes up, it loads, hopefully. Down here, you can rotate. Now, in some graphics programs, to rotate about Z, you have to drag the mouse at the bottom, but nowhere does it tell you that. So even something like that. You then go and you can choose the background, you can choose the style simply by clicking buttons. You don't have to remember the commands. You can color and hide residues, no command line to write. You simply, as if you're selecting a calendar booking for your flight. And you can rub out when you've made mistakes, or you want to just highlight part of the protein. You can add in the side chains again. Your sequence is there, you don't have to remember. You also haven't got the problem of numbering, because the PDB, you can label the residues you want with a variety of styles. And here you're colouring in surfaces. This is very good if you want to highlight a genetic variant. You can label residues. You've got a choice of various forms, 1-letter code, 3-letter code. And then you can click and you get an image you can save. So that's it. That's what you can get. So that's Easymode. If you type Easymode into Google, hopefully you'll get to our site. So the team so far, Lawrence Kelly is the driver for Phyre. Stefan's been working on it. 3D ligand sites, the work from MarkWass at Kent. Suspect is Chris Yates. You see there's no university here. He's now using machine learning for gambling. Easymode is the work of Chris Reynolds. And we've got have we got a minute or two?
You've got a couple of minutes, Mike. Yep.
Perfect timing. So a bit of fun. Bioblocks, it's a protein docking game similar to the idea of fold it, and there's a variety. There's Bioblocks 3D, which we're aiming to make as the scientific tool. The aim is to do crowdsourced protein docking. You'd have the challenge of working out how two proteins dock. Based on that, we said, well, this is quite fun. We've developed a VR version where you've got a nice headset, and that's great for museums. And then, this is in collaboration with Goldsmith, we've got a Tetris-type game that you can download from Apple Play or the App Store, Bioblocks 2D. It works and it's free. And so this is the sort of collection we have. So this is the docking game. You'd start with your two molecules and you'd be able to inspect them and sort of work out where you think they might pack one against the other. You then click on residues, and then when you're interested, you can pull them together and you get a score if it works. You've got a variety of representations you can use. This is the VR version. You put on a headset and you really get a look. You can look into the molecules in a variety of representations. This is more for fun than for docking, though it's interesting how much VR might help in visualization. Anyone wants to discuss that afterwards, I'd be interested, because that's an interest of ours. And this is the sort of Tetris game, which is based on docking ligands into the receptor. You've got the chargers, so it's more complicated. You've got fragment-based drug discovery. You've got to choose the fragments that match. And we've linked this in with a scientific quiz. And I hate computer games, but this is quite good fun if you're on the London Underground. And that's freely available for download, so please try it, and the other games, and send us your comments. And this is a collaboration with Goldsmiths College, particularly Frederick Fowler, Marie, William Latham, and a large team of people more involved in computer games than I am. And that's it. Thank you very much.
Thank you very much, Mike. So we can take one question while we're just changing over the projecting computer. Has anybody got a question for Mike? Thoughts and comments? I think, Mike, the game you put up at the end there looks like a lot of fun. Looks like a lot more difficult than Tetris as well. Yeah. So, okay. Well, please feel free to chat to Mike afterwards. I'm sure Mike will be here for a couple more days. Okay, so just hang on a second. We're just switching over projection.